Black pod disease. Black pod disease is a protozoal disease of cocoa trees. This pathogen, if left untreated, can destroy all yields. Annually, the pathogen can cause a yield loss of up to 1/3 and up to 10% of total trees can be lost completely. With the value of the cocoa industry throughout the world being so large, there are much research and control efforts that go into these Phytophthora spp pathogens. This pathogen can be located anywhere on the cocoa trees, but is most noted for the black mummified look it will give to the fruit of the cocoa tree. Staying ahead of the pathogen is the best means of control. The pathogen can be greatly reduced if leaf litter is allowed to stay on the ground, and if the pathogen gets out of hand, chemical control can be used. This pathogen is mostly found in tropical areas where the cocoa trees are located and need rainfall in order to spread its spores. Doc. Disease cycle, symptoms and signs. The symptom of black pod disease is a necrotic lesion on the cocoa pod with brown or black color, which eventually enlarge to cover the whole pod. White mycelia growth on lesions that appeared several days after infection is a sign for the causal pathogen of black pod disease, which is Phytophthora spp. Black pod disease starts when the infected pod shows some little yellow spots, which eventually turn brown and enlarge to a dark brown or black lesion within 5 days. The lesion is fast growing and covers the entire pod after 8 days of infection. The infection does not only occur on the pod surface, but also invades inside the pod, affecting the beans. The growth of white mycelia on black pod is visible after 11 days and the sporulation is initiated. The dispersal of sporangia or zoospores to water, ants, and other insects occurs at this stage and will infect other healthy pods nearby. Direct contact of a black pod with healthy pods also leads to the spread of disease. In addition, the infected flower cushion and mummified pods are the locations for P. pomivore survival during dry season, where the pathogen will grow and continue to infect other developing pods. The infection occurs on any stage of pod development, where it causes wilting and dying of young pods and destroyed the beans of mature pods. The fully infected pods, the mummified pod, which then become dehydrated, are capable of providing the inoculum of P. pomivore for at least 3 years. P. megakarya causes the same symptom as P. pomivore, but the occurrence is faster and generally produces greater amount of spores. Both P. pomivore and P. megakarya also cause canker on bark, flower cushion and chupons, and cankers on the base extend to the main roots. Cankers were identified as one of inoculum sources for black pod disease. Furthermore, the pattern of infection caused by P. megakarya starts from the ground and moves up to the canopy. However, there is no distinct pattern of disease infection caused by P. pomivore was reported. This pattern of infection could be due to P. megakarya and P. pomivore that were found to survive in soil, and P. megakarya could be surviving in the roots of a few species of shade trees found in cocoa plantation. Hosts. Black pod disease, while its name indicates that it is found in the bromocaca, more commonly known as cocoa trees, also has different hosts. For example, P. megakarya has been detected in the roots of shade trees of Western Africa. While these trees can also be affected by the pathogen, it is because of the market value of the cocoa trees that all emphasis and research on the disease is done on cocoa trees. Cocoa trees. In the cocoa trees, P. megakarya infects the bark, flower, and trees with cankers. These cankers will often exude a reddish gum, reducing the life of the tree, in turn reducing the yield of the plant. The most devastating place the pathogen attacks is in the flowers, as from these flowers is where the cocoa fruit will set. An infected flower will have infected fruit, which will turn black and will be unable to be harvested and processed. Pathogen. 7 different pathogens have been named to cause black pod disease all over the world. All of the pathogens are found in the genus Phytophthora, plant-damaging oomycetes. The 7 species responsible for black pod disease are P. cactusi, P. citrophora, P. megaspirma, P. katsuri, P. pomivora, and P. megakarya. While all of these pathogens can cause black pod disease, the 2 major pathogens are P. pomivora and P. megakarya. The black pod disease pathogens affect all cocoa trees all over the world. P. cactusi and P. citrophora are found in Central and South America, while others such as P. megakarya is found in Central and Western Africa. All of these pathogens can present themselves in all parts of the cocoa trees, but are most devastating in the fruit itself, where it will turn the fruit to a black mummified casing. This fruit is then useless and can no longer be used. Environment. P. megakaryotes found in Central and Western Africa. During the cooler, wetter times of the year, there is a spike in the incidence of black pod disease as when compared to the hotter, more dry times of the year. The disease has a spike shortly after rainfall. The humid weather that is associated with this pathogen and all other black pod disease pathogens is needed as the sporangi forms and starts distributing spores through rainfall, splashing water, and running water. With this pathogen spreading behavior needing water is why there is almost always a spike in the prevalence of the disease shortly after the rainy season. Soil and litter. The spread of infection to pods above bare soil was shown to be greater relative to pods above litter. The reason for this is due to the splash of rain from bare soil spreads the inoculum to pods. However, litter under the tree prevented water droplets from splashing the soil particles as well as the inoculum beneath the litter to the above pods. Further, it was also reported that pods near the ground showed greater infections compared to higher pods due to greater soil was splashed and stick on the lower pods, therefore causes more infections. Air and water dispersal from spore-relating pods. The spread of spores through air from infected pods was observed, and some assumption regarding this mechanism of dispersion has been made in previous studies. However, it is remained uncertain due to unconvincing results from an experiment that collected some spores in the air using the volumetric spore trap, where a small amount of spores were found in the trap. Conversely, the dispersal of inoculum via rain is considered an effective mechanism in spreading the inoculum. It was assumed that under a close canopy, less water will reach the spore-relating pods to spread the inoculum. However, rain dropped from leaves and branches could also splash the inoculum to the surroundings. Infected pods lying on the ground or litter could also spread the inoculum, yet greater infection was observed on pods located under infected pods hanging on the tree compared to pods at the same level of infected pods. It was reported that splash of wind-blown droplets from the infected pods are also able to infect pods on different trees nearby. Invertebrate. The dispersal of the disease is also associated with the invertebrate vectors. Tent-building ants such as Chromatogaster striatula and Campanota zygopymensis were reported as the primary vector in disseminating the spores of P. Phytophthora from infected pods to healthy pods in Ghana. Celsius. Striatula was thought to be the most important vector that is responsible for black pod losses due to its building tent behavior as well as its dominancy within the area under particular condition. In Ghana, Celsius. Striatula removes the outer layer of cocoa pod and uses this material to construct the tent. Therefore, this ant effectively spread the disease by transporting spores from the infected pods on the ground or on trees to healthy pods. Several other ant species, namely Celsius, Africana Celsius, Clairventris in Celsius, Depressor, were also responsible for the spread of the disease besides Celsius. Striatula, in addition, Campanota zygopymensis, another type of tent-building ant that uses soil as building materials for tent construction, was identified as the most important agent to spread the inoculum in Nigeria. Soil was identified as the source of inoculum for P. megakarya, and therefore it was implied that this ant species might use the infected soil to build tents, which infect healthy pods on trees. Other invertebrates that were reported to be associated with spreading the disease are several species of beetle, snail, caterpillar, and millipedes. Fecal samples from these organisms were found to contain viable spores of P. Phytophthora. It was reported that beetle of family Nittigelidae is the most common vector for a black pod, as it was found 50 to 60% of black pod occurrences. These invertebrates consume the outer layer of the infected pods and incidentally ingest the mycelium and spores of the pathogen, thus spread the pathogen to other healthy pods. Disease management. There are several methods available in order to control black pod disease, such as cultural, chemical, and biological control. In addition, the cultivation of varieties that are resistant to black pod is an alternative in order to reduce disease incidence. Cultural control. Several cultural practices to manage black pod disease could be implemented in cocoa plantation. A spacing of 3.1 x 3.1 when pruning of trees are recommended for cocoa planting in order to allow more light and air flow around the trees. This will reduce the level of humidity that is causing black pod disease. The removal of pods with black pod symptoms should also be done in favor to eliminate the source of inoculum. In another study, the utilization of litter mulch under cocoa plantation has been reported in Papua New Guinea, which has some negative effect on the population of P. pomivora and therefore could reduce the pod infection, especially at the beginning of raining season. Leaf litter showed rapid decline in pathogen recovery of colonized cocoa tissue after 18 weeks, relative to grass ground cover. An explanation for this is due to higher moisture content and microbial activity of other microbes under leaf litter that reduces the survival of Phytophthora cinnamomum, as documented by Oriant et al. 2000. More frequent ripe pod harvest, i.e. twice a week, and removal of infected pod on the ground was demonstrated to significantly reduce disease occurrence and improve pod yield compared to less frequent harvest and removal, i.e. once a month. In addition, scattered healthy pod on the ground should also be removed, as it will be infected and become the source of inoculum later. Sanitation is one cultural method to control for black pod disease. Sanitation practices include weed removal, pruning, thinning and removal of infected and mummified pods every 2 weeks in order to eliminate the source for inoculum. Phytosanitary pod removal was observed to significantly reduce disease occurrences by 9-11% to 22-31%, where this practice removes the source for a secondary inoculum. However, increase in disease incidence after raining season was observed to be most likely due to the spread of inoculum from survival site by the rain. The application of fungicide following sanitation is commonly performed for an effective control of disease, as sanitation practice alone would not eliminate the source of inoculum and still causes greater black pod incidence compared to sanitation followed by at least one fungicide application. Chemical control. The application of copper fungicide has been shown to significantly reduce a great number of black pod incidences in Nigeria. Metalaxyl-ridamil and cuprous oxalate perinox were identified to be successful in increasing the number of harvested healthy pod compared to the application of fused aluminium halide and control treatment. On top of that, the timing of fungicide application has some positive effect on the final pod yield, where this plot produced greater yield than the unsprayed plot. The application was done before August, which is before the main disease epidemic that usually occurs in September and October. The recommended standard for fungicide application to control black pod disease caused by P. megakarya for a season is 6 to 8 times of application in every 3-4 weeks. However, the adoption of recommended application was very low among farmers in Ghana. Therefore, an experiment with a reduced number of fungicide applications demonstrated that there was 25 to 45% reduction in disease incidence. In terms of disease control and yield, sanitation and 3 applications of ridamil 72 plus 12%, metalaxyl plus 60%, copper minus 1 oxide fungicide showed a better control compared to sanitation alone and sanitation with 1 or 2 fungicide applications. However, reduced in fungicide application was shown to be significantly less effective than the recommended standard fungicide application. It was suggested that the understanding regarding the source of inoculum, the amount of infective inoculum production, and how the disease is disseminated is important in order to identify the appropriate and economical method in fungicide application as well as for an effective control of the disease. For example, the application of fungicide on the trunk will help farmers to control the spread of the disease up in the canopy, as it is difficult to reach the canopy during fungicide application. This will eventually save more time, labor and cost for disease management. Cultural and chemical control. In Ghana, a study that combined the sanitation and fungicide application showed a significant reduction in the percentage of disease incidence, where greater black pod incidence were observed from pods on the trunk in the canopy and control treatment no fungicide application. This suggested that the application of fungicide on the trunk would protect pods from infection, therefore reduce primary and secondary infection rate both on the trunk and in the canopy. In addition, the application of systemic potassium phosphonate with 1 and double injection 20 ml and 40 ml of fungicide for each injection frequency and semi-systemic metalaxyl fungicide showed better control compared to contact fungicides copper-based fungicide in both locations that were used in the experiment. Biological control. Heavy application of chemical fungicide would eventually lead to resistance of pathogen and causing soil and water pollutions. Hence, more sustainable and environmental friendly methods should be established and implemented, such as biological control. Several species of fungi from the genera of Trichoderma, was identified to be a beneficial endophyte to control black pod caused by Phytophthora spp. An isolate of Trichoderma spirellum from soil was observed as a potential mycoparasite for P. megakaryote, where this fungus has the potential to reduce black pod incidence under field condition in Cameroon. It was reported that moderate black pod cases 47% occurred in the T. Spirellum treatment to control black pod disease compared to trees with untreated 71% and chemical fungicide 1.73%. Another species, which is T. vians, also has been documented to reduce on black pod incident in Peru. In Brazil, a new species known as Trichoderma Marshall Samuels sp. Nov. was identified as an endophyte on cocoa, which has the ability to reduce black pod symptoms caused by P. megakaryote. This endophyte species survives on cocoa pods and has the ability to establish a long endophytic association with the host about 3.5 months. Nevertheless, the protection against black pod via biological control is not as effective as the control using chemical fungicides. Resistant variety. There is no specific variety of cocoa that shows resistant to Phytophthora infections, and the establishment and utilization of resistant variety will be most likely depends on the region. Numerous breeding programs have been established worldwide in order to screen and test for local hybrids for disease resistant to Phytophthora spp. For example, a study in Cameroon assessed the performance of local cocoa cultivars, the southern and northern Cameroon cultivar, compared to the local and international gene bank cultivars. The local Genebank cultivar consisted of foreign hybrid of Upper Amazon X Trinidad, and an international cultivar from Papua New Guinea and Latin America were provided through International Cocoa Genebank Trinidad. Based on the information provided by farmers and leaf disc tests to assess resistant variety, the local cultivars selected from farmers' field showed some resistance to P. megakaryote compared to other varieties. Thus, it was concluded that there are some potential resistant varieties available in this area. In addition, some molecular work on developing resistant varieties to black pod is being done by CEPLAC Executive Plan of Cocoa Farming Agency in Brazil, and hopefully more breeding program focusing on black pod resistant will be established and produced commercialized resistant varieties. Importance. The United States chocolate industry consumes $1.4 billion of cocoa and supplies 68,450 jobs throughout the United States. With this industry being so important not only in the US but also all over the world, the black pod disease is of high importance. This pathogen, if left untreated, can destroy all yields. Annually, the pathogen causes 20–30% yield loss and kills about 10% of trees completely.